Hello, everyone. I'm Linda Tintawi, CEO of the Lustgarten Foundation, and welcome to the first installment of LustgartenLIVE for 2023. We have the largest registered audience ever since we launched these live stream events in 2021. Nearly 300 of you from across the United States and Canada want to know more about Lustgarten's landmark and timely partnership with Breakthrough Cancer. We encourage you to ask questions. You can use the Q&A box at the bottom of your screen, and we'll answer them at the end of today's session. Today's live stream event will be recorded and available on lustgarten.org's website, lustgarten.org/live shortly after today's event. Now on to our program. Dr. Tyler Jacks is a world leader in the field of cancer genetics and is known for his groundbreaking work on the development of genetically engineered mouse models of cancer. Dr. Jacks is the David H. Koch Professor of Biology at MIT, is director of the Blue Ribbon Panel for President Biden's National Cancer Moonshot Initiative, the head of Lustgarten's dedicated lab at MIT, a member of the Lustgarten Scientific Advisory Board, and of course, president of Breakthrough Cancer. It's now my great pleasure to bring you Dr. Tyler Jacks.
Thanks very much, Linda. It's a real pleasure for me to participate in this exciting program. As you've heard, I've had a long affiliation with the Lustgarten Foundation dating back more than 20 years, actually. I've been an advisor to the Lustgarten Foundation. I'm still a member of the Scientific Advisory Board, and I've received support from the Lustgarten for research in my laboratory on pancreas cancer for many years now. And I'm currently the proud head of the Lustgarten dedicated lab at MIT for pancreas cancer research. And so deep connections to the Lustgarten Foundation and great respect for the organization. In addition, I'm the president of Breakthrough Cancer, which is a new foundation partnering with the Lustgarten Foundation on pancreas cancer research. Breakthrough covers other cancer types, but as it relates to pancreas cancer research, we are pursuing these projects collaboratively with the Lustgarten Foundation, and you're going to hear about two of them today. Briefly, the Breakthrough Cancer is a foundation that was created a couple of years ago. It brings together 5 very strong cancer centers across the country. MIT, Dana-Farber Cancer Institute, Memorial Sloan Kettering Cancer Center, Johns Hopkins, Kimmel Cancer Center, and the MD Anderson Cancer Center. Great expertise across these organizations, and with funding and organizational help from Breakthrough Cancer, we hope to bring out the best from these institutions to tackle some very challenging cancer types, including, importantly, pancreas cancer. And we want to bring a sense of urgency and to demonstrate the value of teamwork and collaboration as we execute these projects. Lustgarten has been an awesome partner in the pancreas cancer projects really since day one, sort of grounded by our many mutual connections, mine and Dave Tuvison, but also many of the investigators with whom we are working in this project have had their own connections to the Lustgarten Foundation. So it just made sense for us to work together. We believe at Breakthrough Cancer in what we call radical collaboration. That's collaboration that cuts across institutions, cuts across individuals, cuts across organizations. And this is a perfect example. 2 foundations coming together to pool resources, to carry out some big and important projects collectively. We're also interested in reaching out to industry, and you'll hear a bit about this in the presentations today about how important it is that we can work productively with companies that are producing some of the drugs that need to be tested or technologies that are being perfected in the hands of those companies. So these are exciting times between Breakthrough Cancer and the Lustgarten Foundation. And this is going to be an exciting program. Really at the core of the projects are the scientists that we are supporting, the clinicians that we are supporting, the engineers that we are supporting, and all of the investigators who are participating in the projects. And you're going to hear from 2 outstanding investigators today. Laura Wood from Johns Hopkins working on one of our 2 projects using very cutting-edge molecular analysis tools to really understand what's happening in cancer patients undergoing treatment. and also Andy Aguirre, who is leading the team involved in understanding how we can drug the KRAS protein, which is implicated very strongly in the development of pancreas cancer. And you'll hear a lot more details from both Laura and from Andy. So without further ado, I'll turn over the program to Andy Rakeman, who will introduce Laura and Andy. Thanks so much and enjoy the program.
Thank you so much, Tyler, and thank you, Linda, for kicking us off. And it's so exciting to be here for this event, and in a lot of ways not surprising to me, at least, to hear that this is one of the largest registrations we've seen for one of these events, because I think we have a lot of very exciting things to talk about. We're going to talk about some really very exciting science, some science that has real promise for transforming what's possible in pancreatic cancer, but also, exciting for me, from my position as VP of Research here at Lustgarten, you get a little bit of a behind the scenes view on what does it really take to get this science going and to show progress there. And really at the center of how we'll talk about that today is this partnership between the Lustgarten Foundation and Breakthrough Cancer. And as Tyler mentioned, Breakthrough Cancer is a relatively new foundation, a relatively new player coming into the scene here. And when we heard about this foundation starting, and that it was going to be this group that's really focused on taking on the hardest cancers, the toughest cancers out there, was going to work in a way where it could build together these teams that were going to address the most critical questions and the toughest questions in those cancers, and the ones that are blocking us from getting to where we need to be in terms of patient outcomes, and doing that in a way using this radical collaboration concept, right? A way that really knocks down the barriers that sometimes slow progress or make things a little bit difficult. We at Lustgarten said, we're all in, that's how we want to go. And also recognize that there was going to be a lot of potential for us to do a lot here, knowing that pancreatic cancer is one of the toughest cancers out there, that there are challenges that are preventing us from having the impact we need to have in patients, but that the solutions to those are starting to come into focus. And really, because of this 25-year history of supporting people, bringing out the teams, getting the fundamental knowledge about what drives this disease, building out the tools to really understand it best, that we were in a position with this kind of partnership to really supercharge progress if we could focus the right efforts on the right questions. And I think you'll hear a lot about that today. So we'll hear two very wonderful presentations from Dr. Aguirre and Dr. Wood about the very exciting work that's happening in their labs, and as you heard, across these 5 institutions participating in the Breakthrough Cancer program. And you'll hear a lot of science and a lot of technical things, a lot of stuff that may even look like science fiction of what's becoming possible with new technologies today. But a few key things that I think undercut all of what you'll hear that'll be good to pay attention to is the idea of collaboration, that these are projects that are happening, multiple funding sources, multiple institutions, multiple expertise coming to bear here. And that's really enabling what's possible with these projects. The focus on patients, I think the focus on patients, both in identifying what are those key gaps that we need to address to have the impact we want to have, but also learning from patients about what is the biology that we're trying to address. Having that humility and humbleness to say we don't know it all. As scientists, we need to learn from the patient as well. And that's going to help us focus on where we need to go, and how that all then builds back into the sense of urgency that you'll hear about from Dr. Aguirre and Dr. Wood, and how these projects are really being supercharged and really trying to move as fast as possible and drive our progress even faster than we've been moving in the past. So I know you didn't come here to hear me. I know you came to hear the wonderful scientists that are joining us. So I'll move on to introduce Dr. Aguirre. Dr. Aguirre is a physician scientist from Dana-Farber. He is a medical oncologist as well as a researcher. He primarily works at Dana-Farber, also has appointments at Harvard Medical School and the Broad Institute at Harvard and MIT. So sees patients in clinic, but also runs a basic and translational research laboratory where he focuses, among other things, on really understanding pancreatic cancer, and in particular has really emerged as a leader in understanding the role of RAS and these new RAS targeting drugs and their potential in treating cancers and their potential hurdles in the future, and how resistance can come up to these drugs going forward. So I'm sure you'll hear from Dr. Gary. RAS, as many of you know, is kind of the smoking gun of pancreas cancer. It's the driver of most patients with pancreas cancer. And what we're really trying to do here is turn it into the Achilles heel of pancreas cancer, and really a way then to use it not only as a liability, but as the way to treat the cancer going forward. And so thank you so much for joining us, Andy, and I really look forward to hearing about what you see as the key hurdles and the promise and the excitement around RAS right now, and how this team is really poised to take that on and bring that forward.
Well, great. Well, thank you very much, Andy, for the opportunity to speak here. Andy and the entire Lustgarten Foundation, Linda and colleagues, it's really a privilege to be here. Thank you to Dr. Jacks and the colleagues at Breakthrough Cancer as well for the opportunity to represent them in this project in this forum. I'm just going to share my screen quickly and we can get started. So as Andy mentioned, I'm a medical oncologist at Dana-Farber Cancer Institute, and I run a research lab focused on pancreatic cancer. And I've had the great privilege of working with the Lustgarten Foundation over the course of almost 20 years now, since I was a student in medical school. And then throughout my career, they've provided enormous support and a community of researchers and clinicians focused on pancreatic cancer. So it's really been an anchor for my own training and a great source of support throughout my career. So it's a privilege to speak in this forum. And it's also a great opportunity to talk about something that's one of the most exciting opportunities, I believe, in my career and in making a difference for pancreatic cancer patients, which is targeting the KRAS oncogene. And so the goal of this presentation is to go through a little bit of the background of why KRAS is so important in pancreatic cancer, but also to give you a sense of the problem and a reason for optimism, and that hope is here, and we have a lot to look forward to in how we may change how we treat pancreatic cancer patients in the future. And I just want to start by acknowledging that I'm a representative of a large, diverse group of colleagues that we've termed the Conquering KRAS and Pancreatic Cancer Research Team. And it's a collaborative team funded both by the Lustgarten Foundation and Breakthrough Cancer. And I'll highlight some of those members in a bit. So I just wanted to start by telling the audience about the typical care of a pancreatic cancer patient today. Many of you on the call may know this all too well and have had experience with family members or even yourself, or take care of patients with pancreatic cancer. But this is a case of a 49-year-old woman who presented to her primary care physician with 6 months of weight loss and some mild abdominal pain. Unfortunately, was diagnosed with advanced pancreatic cancer that had spread beyond the pancreas. We typically get clinical sequencing to try to aid our understanding of the cancer, the type of cancer, how we might treat a particular patient's tumor. And all too often in pancreatic cancer, we get results such as this, that there's a KRAS mutation and it tells us what won't work in that particular patient. And there's no indication of what will work or how we can leverage that KRAS mutation to better treat patients. And unfortunately, over 90% of pancreatic cancer patients have a KRAS mutation. And for a long time, it's been a real challenge in the field, and how we treat patients is to have this clear driver of pancreatic cancer present in the vast majority of pancreatic cancers, but not be able to do anything about it. And standard treatment options have come a long way. And through the support of organizations like the Lustgarten Foundation, the improvements have been made in how we treat patients with standard chemotherapy. And this really represents one of the best standards of care for pancreatic cancer patients, a multi-agent regimen called FOLFIRINOX compared to single-agent gemcitabine. Clear benefit to these multi-agent chemotherapies, especially in certain subsets of patients. And this has led to really a doubling of the overall survival in the last 5 to 10 years using these modern regimens and the median overall survival is about 11 months. Now that's an improvement from where we were a decade ago, but still not good enough. An 11-month median overall survival is, it leaves significant room for improvement. And until recently, there really have not been what we term targeted therapies, molecularly driven therapies, maybe often given in the form of a pill for the majority of pancreatic cancer patients. And I'm going to highlight some recent work from a vast group of colleagues and pharmaceutical companies that really is changing that statement currently. And so, as I'd mentioned, over 90% of pancreatic cancer patients have a mutation in the KRAS gene. It turns out KRAS is the most frequently altered oncogene in human cancer, the most frequently altered gain of function sort of driver of cancers worldwide are mutations in the KRAS gene. And unfortunately, these KRAS mutations that occur in over 90% of pancreatic cancer patients contribute to 45,000 pancreatic cancer deaths each year in the United States, a staggering number of patients dying from pancreatic cancer and driven by these mutations in this RAS oncogene. And it turns out we've known about RAS for decades now. It was really the first human oncogene that was identified by work from Bob Weinberg and colleagues at MIT and Merck Laboratories, as well as at the National Cancer Institute, identified that RAS mutations occur in human cancer. And an enormous body of work has since occurred studying RAS, that we know where the mutations occur, how they occur, what they do to cancer cells, how they change the signaling in cancer cells. But we've yet been able, not yet been able to do anything about these mutations. And just again, stating pancreatic cancer is really a KRAS-driven disease. This is work from colleagues at Johns Hopkins many years ago now, illustrating that pre-invasive lesions occur in the pancreas. These pre-invasive or pan-in lesions occur, and they go through a series of stages leading to advanced cancer or carcinoma. KRAS mutations are the earliest and most frequent alterations that occur even in these pre-invasive lesions that drive to advanced cancer. And work from a number of colleagues, but really pioneered and led by David Tuvison, who's the chief scientist at the Lustgarten Foundation, and Dr. Jackson colleagues, Dr. DePino and others, studying RAS mutations in genetically engineered mouse models of pancreatic cancer that are mice are engineered to get pancreatic cancer. What we know is that when you give a mouse a KRAS mutation in the pancreas, it develops pancreatic cancer. Similarly, if you take that KRAS mutation away in advanced tumors in these mice, you can shut it off genetically. Those tumors melt away and contribute to prolongation of survival of these mice. So clear evidence RAS mutations occur early and often in pancreatic cancer. You can generate pancreatic cancer in preclinical models by giving RAS, and when you shut it off, that can lead to regressions of those tumors. So we've learned a lot about the mechanisms of cancer formation from RAS over the years. KRAS normally transmits signals from outside the cell inside the cell. It takes growth factor signals and transmits them into signals for the cell to grow and survive. And it acts as what we think of as a relay switch, that in one state it's on and signaling, in another state it's off. And it turns out that mutations in KRAS, as occur in human cancer, these alterations to the gene, which lead to alterations in the protein, they lock the KRAS protein into an on state and cause signaling that's constitutively on in the cancer cell and really driving progression of the cancer cell. Well, it turns out that RAS does a lot of things in the cell, and it activates multiple molecular pathways. And this is intended to be a complicated diagram to show you that RAS does many things, but in particular, it drives this orange signaling pathway here, the RAS MAP kinase signaling pathway, but also activates a number of other pathways. And drugs have been developed to target various members of these pathways, in particular, this orange signaling pathway. Unfortunately, even though these are very good drugs, the cancer cells adapt to these drugs. And so while they may work for a short period of time, the cancer cells adapt. and develop mechanisms of resistance. Moreover, there are toxicities to some of these drugs. These drugs target proteins that are also active and important in the normal cell. And so while they can be effective in preclinical models, toxicities have really limited how we use these in patients and have really thwarted their use in pancreatic cancer. And this model here represents sort of a spinning diagram of the RAS protein itself, the structure of the protein. And it turns out that mutations in RAS actually alter the structure of the protein. It changes how it looks molecularly and structurally, and changes how it functions. Unfortunately, it's been very difficult to develop small molecule inhibitors, the type of inhibitors or drugs that you take by mouth as a pill form. It's been very challenging to get those molecules to stick to the RAS protein and to inactivate that activated cancer-causing form of the protein. And RAS has this highly dynamic structure, changing in these flexible regions. While it binds a nucleotide, and sometimes in certain other types of proteins that are altered in cancer, we can outcompete nucleotide and create inhibitors. It has the RAS protein has an extremely high affinity for this nucleotide, so it's virtually impossible to outcompete it. So it's been really a challenge to develop drugs that target the RAS protein. But there's been an enormous amount of work, decades of work, to understand the biology of RAS signaling done throughout academic laboratories, pharmaceutical companies and industry partners, as well as at the National Cancer Institute. And about 15 years ago or so, the National Cancer Institute really rejuvenated a focus on developing drugs to target RAS. And this is called the National Cancer Institute RAS Initiative. And it was a kind of hub and spoke model, centrally focused at the National Institutes of Health, but funding laboratories throughout the country to work on RAS, to focus on RAS, developing industry partners and partnerships with small molecule inhibitors and drugs and pharmaceutical companies to really bring the focus back to target this protein and really attack this difficult challenge head on. And then there have been enormously important scientific discoveries, and I highlight one in particular here. Colleagues from the University of California, San Francisco, Kevan Shokat and colleagues, as well as a number of pharmaceutical companies, developed new small molecule inhibitors that bind in a protein pocket that was previously underappreciated, that this pocket existed, but found molecules that could covalently or irreversibly bind to this pocket and to the RAS protein. And this led to a revolution in drug development, that there were now multiple small molecule inhibitors developed by multiple different pharmaceutical companies that could bind to this pocket and inactivate the RAS protein. And there's been some early clinical experience with these inhibitors in pancreatic cancer patients. And this is a summary of a significant amount of data in 48 patients with 2 different drugs. Not always a great thing to do to compare across drugs in different studies, but I wanted to highlight the potential for these inhibitors in a simplified form. 27% of patients treated with one of these inhibitors had a response to therapy, as indicated on the left here in this pie chart. And in terms of disease control, in terms of patients who either had a response, their tumor shrunk with the drug, or they had stable disease. They were able to continue to live their lives, their cancer not growing for a period of time. Almost 90% of patients in aggregate had stable disease. So profoundly important results. These were patients that had been treated previously with chemotherapy, advanced, typically metastatic disease, and in a setting where standard treatment agents such as chemotherapy maybe only gives a 2 or 3% response rate. These drugs were now coming in and giving a response in up to a quarter or a third of patients. So profoundly important in this advanced setting, that some evidence of signal and responses and clinical benefit for pancreatic cancer patients treated with these drugs. And it turns out this is really just the tip of the iceberg. There are multiple different RAS inhibitors that are on the horizon in drug development pipelines, in pharmaceutical companies. Many different RAS inhibitors coming to the clinic in clinical trials. So we spoke about this adagrasib and sotorasib. These 2 drugs are now approved for use in lung cancer, and there are the clinical studies ongoing that may ultimately lead to an approval in pancreatic cancer. And the results really in the FDA submissions, and we're awaiting to see if these can indeed be approved for use in pancreatic cancer patients. And these are specific for a particular mutant form of the protein that occurs only in a small fraction of pancreatic cancer patients. Only in about 1% of pancreatic cancer patients have that flavor of KRAS mutation. But there's reason for optimism that a number of other mutant forms of the protein are also targetable with new drugs, including the KRAS G12D mutant form of the protein, which is the most common mutation that occurs in pancreatic cancer, KRAS mutation that occurs in pancreatic cancer, and similarly many other RAS mutations. In fact, there are drugs developed that target all RAS mutations and all RAS isoforms, all different types of RAS. And these are entering clinical studies and in patients currently. So tremendous reason for hope that these drugs are going to reach more patients and expand the impact and the importance of KRAS inhibition in pancreatic cancer patients going forward. And so we've recognizing this, that hopefully in 3 to 5 years, all pancreatic cancer patients may have access to drugs that target the RAS protein. A group of us throughout the country really realized that we needed to focus on what these drugs were doing in pancreatic cancer patients and in preclinical models, and bring the focus on how to best use these drugs to benefit pancreatic cancer patients. And so it was really a tremendous opportunity to take the LustGarten Foundation, which has been dedicated to pancreatic cancer research for years now, and a relatively new foundation, Breakthrough Cancer, which is committed to making a major impact as quickly as possible and bringing the resources and the collaborations to bear on a major problem such as pancreatic cancer. And so we formed this team that we've called the Conquering KRAS and Pancreatic Cancer Team, with the support, encouragement, and funding from the LustGarten Foundation and Breakthrough Cancer, to really keep the focus on pancreatic cancer. While these drugs could be developed in many different areas, many different other types of cancer, let's make sure they're getting developed in pancreatic cancer patients. To understand how these drugs are working, what's their mechanism of action, how do you become resistant to these drugs, and when they're resistant, how do you develop appropriate therapies, combination therapies that target that resistance to get maximal efficacy, maximal impact of these drugs in patients, and then really to develop a robust academic and industry collaboration to benefit patients. This has always been an important focus of the LustGarten Foundation, to nucleate important clinical trials, bring together industry partners with academics, and really a key mission of Breakthrough Cancer, to break down barriers beyond academic and industry and bring people together focused on the right problem and bringing them the right resources to bear on these problems. And there are really a few major goals of this project: to use preclinical models to test new compounds and understand as much as we can about biomarkers of response and resistance, to develop new therapeutics, and ultimately to get those into patients through really well-designed clinical trials that are designed to get an answer quickly and impact the most patients as possible. And so we really thought to bring together experts across many different disciplines: biologists, computational biologists, those of us who study the tumor microenvironment, applying new technologies to understand human tumors and understand RAS inhibition, and of course, importantly, clinical development, experts in clinical trials, and how do we run trials in the optimal way to answer important clinical questions. So we brought together a multidisciplinary team across the 5 institutions that Dr. Jacks mentioned, and across many different disciplines. There are examples of computational biologists, engineers, clinicians, experts in KRAS biology, experts in pancreatic cancer biology. And again, I speak on behalf of this entire group. An enormous think tank occurred to think about what are the most important aspects of this project that we should be focusing on, and how do we best use these new drugs to benefit patients. And how do we leverage the latest advances in technologies in cancer models developed directly from patients' tumors, patient-derived organoid models, or mouse models of pancreatic cancer that the LustGarten Foundation has funded and championed for years. new tumor profiling technologies, such as single cell RNA sequencing or spatial profiling? How do we look not just at the tumor in bulk, but every cell in the tumor and understand how each of those cells is going to respond or not respond to particular KRAS inhibitors, or how do cells communicate or interact between each other? How are fibroblasts or non-tumor cells talking to tumor cells and telling them to grow and survive? And can we cut off those signals to have efficacy in pancreatic cancer? So really an explosion of development of new technologies in the last 5 years even. We're bringing those technologies to bear on this problem. And so really our goals are to understand adaptive responses to therapy. When we give a RAS inhibitor, how do tumor cells react and what signaling mechanisms get upregulated both inside the tumor cell, but also in the tumor microenvironment, crosstalk between non-tumor cells and tumor cells within the tumor itself. And how do we understand those to really develop new combination therapies? Can we identify new candidates, new targets that we should be developing additional drugs toward? And can we combine those with RAS inhibitors and validate those in these valuable preclinical models that we've already developed and now are poised to use? And lastly, how do we take these new therapeutic strategies? We know that KRAS inhibition alone is not going to be enough to really lead to long-term remissions or even cures in patients. But what else do we combine with it? And how do we develop trials that are well tolerated by pancreatic cancer patients and lead to important efficacy and prolong people's lives? And so this project really focuses on all three dimensions here: understanding these at a basic science level, developing the right therapies with the right biomarkers so that we can stratify patients into therapies that they'll benefit from, as opposed to ones that they're not going to benefit from and just get toxicity from. And then how do we use these in patients? And so a fundamental foundation for this project is learning from patients. Every patient that we give a RAS inhibitor to, we're going to learn through biopsies or blood sampling, and we're going to do deep molecular characterization of the patient's tumor. And this is really a collaboration with the patient who goes through these trials, consents to these biopsies and these protocols, and helps us learn best how to use those drugs, not only in that particular patient, but in future patients. And that's really a commitment by patients to commit to helping us learn how best to develop new therapies. And this will be an iterative process. The things we learn from patients will go back into the lab. The things we learn from the lab will ultimately, hopefully, make it to patients and feed into the development of next generations of trials. So we think we're poised for a major impact through this project. We have the right foundations backing us who not only know pancreatic cancer, but know how to bring the right partners together, industry and academia, with a goal to improve patient care, perhaps serve as a paradigm for industry collaboration. How do we bring companies together who fundamentally want to benefit patients, but also have business goals in mind? How do we bring them together with the clinicians and scientists, technology developers, to answer the appropriate questions to develop the right trials for patients? And hopefully this can serve as a model for other high priority targets. We know about RAS. RAS is important. There are many other genes that are also altered in pancreatic cancer, perhaps equally as important to the biology of the disease. For instance, the p53 gene. What do we do about that? And how do we tackle that problem? Hopefully this project, beyond making a major impact with RAS, is going to pave the way for us to focus on other alterations and other problems in a similarly concerted manner. So with that, I'll close and just really highlight again that this is a massive effort from a number of tremendous collaborators and contributors across multiple different institutions. In particular, thank the Breakthrough Cancer team led by Tyler Jacks and Jesse Bohm and a number of great colleagues there. And of course, the LustGarten Foundation for their ongoing commitment to pancreatic cancer research. Andy Rakeman, Dave Tuvison, Linda Tantawi, and others. and their commitment to this project and their collaboration. It's really been instrumental to driving this project forward. And with that, I'm happy to take any questions, or if we're deferring questions until after Dr. Wood's talk, I'm happy to do that as well.
Thank you so much, Andy. That was really an excellent overview of the challenge of KRAS, how far we've come, and I think an excellent description of how this team is really poised to handle all those challenges that you laid out on how do you move into the clinic, but move into the clinic with eyes wide open and continuously learn and iterate and to continuously improve. So for folks who might have joined late, I'll just remind you how Linda opened this up, that if you do have questions, please type them into the Q&A chat, and we will come back and we'll have a little discussion with Dr. Aguirre and Dr. Wood at the end, and we'll address your questions as they've been submitted. But thank you so much, Andy. I hope people leave that really feeling hopeful that this real KRAS gene that's been kind of the bane of pancreatic cancer research for so long is now looking like we may have some real tools to challenge that. And again, the humility of approaching it, saying that this won't be the end, but it's going to, but we need to be ready to adapt to the new opportunities and new challenges that it will present when these inhibitors are available and start making differences for patients. So thank you very much, Dr. Aguirre. And now I again have the very distinct pleasure of introducing one of the lead scientists from our partnership with Breakthrough Cancer, another fantastic physician scientist, Dr. Laura Wood, who is an associate professor and director of the Division of GI and Liver Pathology at Johns Hopkins School of Medicine. So Laura is a physician scientist, a pathologist, as well as a researcher, and runs a lab. And one of the things I've learned in my scientific career is when the pathologists talk, you listen, because they are the folks that really are looking at the disease itself and really, taking away all of the filters that often as molecular biologists and other types of scientists what we put in the way. So Dr. Wood in her own lab has really been developing some cutting edge tools to bring molecular analysis and 3D analysis and cellular analysis to pathology, to the understanding of disease. And so very appropriately is here to represent the Breakthrough Cancer LustGarten project on demystifying pancreatic cancer. So really developing and deploying these types of tools to really understand the disease. And it's a good segue, I think, from how Dr. Aguirre closed out his talk, really help to close that loop from clinical science and patients back to the bench and forward again, and really make sure that we can learn as much as possible from patients and in a way that's going to be actionable and we can take forward into the clinic again in the future. So Dr. Wood, thank you so much for joining us. And I'm sure you have a lovely presentation and as pathologists do, and as I know as you always do, with some very lovely pictures of what this disease looks like and what we can really learn from it when we take a step back and let it teach us.
Yeah, thank you so much, Andy, for that introduction, and just thank you to all of the folks joining the webinar who have come to learn and hear about our work. I really appreciate the opportunity to present on behalf of our entire team through the Breakthrough Cancer and LustGarten Foundation. And so the title of our project is Demystifying Pancreatic Cancer Therapy, and what we want that to capture is that what we're trying to understand is how different therapies affect the pancreatic tumor, and then how pancreatic tumors develop resistance, and then how we may be able to target that resistance in the future. And so I'm going to give some background and also try to help you understand why we think that's going to be a powerful approach to improving treatment for pancreatic cancer patients. So pancreatic cancer treatment is very challenging, and we have made some improvements over the last years, right? The survival curve has gone up, but we still have a long way to go. And I put this graph on here to highlight, we have moved it in the right direction, but we still have a long way to go, and we need big ideas and new ideas as to how to do this. I put a sample of the results of several recent clinical trials. And unfortunately, many of the results of these trials are disappointing. They either don't improve the survival of patients or improve it only slightly. There's a number of different reasons for this. Preclinical models are very powerful, and all of these treatments seem to work well in preclinical models, but then it doesn't always translate to humans. And we don't always entirely understand why that is. And so the main goal of our project is really to understand in human patients, in clinical trials, why are therapies working or why are they not working? How can we leverage really unique technological resources at all of our institutions to really understand in human patients in the trials, why are the therapies working and why are they not working? And then how can we use that to design better clinical trials in the future? So the first set of trials is focused on localized pancreatic cancer. It provides us some really unique opportunities to better understand the disease. Unfortunately, in pancreatic cancer, only about 20% of patients present with disease that can be removed surgically. So this is disease that's localized only to the pancreas and can be taken out by surgery. Most of the improvements in survival have happened in this population, but still the majority of patients, even with localized pancreatic cancer, so the earliest stage that can be taken out surgically, the majority of those patients still recur. And so this population of patients really represents a unique opportunity for identifying what are the barriers to long-term survival in pancreatic cancer in this population. And we do think that in the long term, the things we learn from this population of patients can be extrapolated to the larger group of patients, even without resectable disease. So what are the pivotal questions that we're trying to learn from pancreatic cancer therapies as we think about these studies? We want to understand how cancer and immune cells interact, right? How do the changes, not in the tumor cells themselves, but the changes in the cells surrounding the tumor, how do those affect therapeutic response? And more specifically, are there specific pathways that the cancers use to interact with the cells in their microenvironment that can make them more responsive or resistant? And then also understanding tumor evolution over time. And again, what changes in the tumor cells themselves can cause therapeutic response and resistance. So we want to understand the tumor cells as well as the cells in the microenvironment and try to understand what changes in both of these are either allowing pancreatic cancer cells to respond to therapy or becoming resistant to therapy. So I want to talk through kind of the general treatment approach to these localized pancreatic cancer patients, and then really highlight why this is going to be a unique way to understand how therapies are working in patients. So a patient who has localized pancreatic cancer is typically diagnosed with a ultrasound guided biopsy. Then they can get chemotherapy prior to their surgery, or therapy as part of a clinical trial prior to their surgery. They then will get their tumor taken out surgically, either with a Whipple surgery or a distal pancreatectomy. Then they'll get more chemotherapy in the adjuvant setting. This means after surgery. And then unfortunately, as I mentioned, the majority of these patients, even who have a cancer that can be resected, they still often recur. And so patients often will have recurrence and metastasis. But with this clinical course comes multiple opportunities, where as part of the clinical care of the patients, we have sampling of their tissue. And what we want to do is to use kind of the most cutting edge technologies that we have access to across our 5 institutions to study these tumor samples that are taken as part of the clinical care of patients to understand better why the tumors are responding and then why they eventually become resistant. And that will let us then design better and new approaches to overcome the resistance. So this is the structure of our platform trial, and I want to acknowledge Lei Zhang, who is one of the clinical leaders in our group who designed this first trial. And what I want to highlight is that the patients get a pre-surgical biopsy, and then they get their first experimental therapy 2 weeks prior to their surgery. Then when they get their surgery, they'll have the bulk of their pancreas tumor taken out. And then afterward, they get their standard of care chemotherapy as well as more experimental therapy. And then we can sample blood over time as well as tumor later. What this lets us do is that we can understand the mechanisms of resistance within this first trial, and then use that to iteratively design the next trial. So we can say that there's a certain cell type or a certain molecular alteration that was important for resistance in this first trial. So let's add in a new agent to then target that in the next trial. And then we can do the same thing down the line, iteratively improving the trials and improving the agents by understanding how the patients in the previous trial became resistant to the therapy. So again, we use the data from the previous trial to inform the design of the next trial. And I really want to highlight that, again, it's iterative combinations, and there is the potential opportunity to have confluence with the KRAS team and to use some of these trial design and tissue profiling techniques to also understand resistance in the KRAS inhibitor trials. So how are we going to use these tissue samples to really understand how and why pancreatic tumors become resistant to therapy? The concept I want you to come away with is that we're going to go both deep and wide. So by deep, I mean we're going to do really deep, what we call 3D spatial multi-omic analysis. I know that's a mouthful, but what I mean by that is we're going to build 3D models of patient tumors, and I'll talk a little bit more about that. And then we can use all different types of profiling techniques to understand how the tumor cells themselves change and how the microenvironment changes. And we can do all of that in 3D in the tumor that was taken out at surgery. So we're going to do very, very deep profiling on that tumor taken out at surgery. But then we also want to go wide. And by wide, I mean looking over time. So we're going to use DNA sequencing and what we call genomic analysis to understand how the tumor evolves over time, because we have this opportunity to have multiple samples from the same patient. So the 3D analysis that I mentioned is something we've developed in collaboration with Denise Wirtz, who's at the School of Engineering here at Hopkins. And the details of exactly how this works are certainly not something we're going to go into today, but what we can do is take a pretty large piece of a pancreas tumor, several cubic centimeters, the size of a stack of quarters, and we can basically make a 3D model and single cell label every single cell in that large piece of tumor. And that can help us visualize and quantify many, many, many different features of that tumor. As Andy suggested, I did come with cool visual aids. This is about 2 cubic centimeter piece of pancreas, or it's actually a piece of normal pancreas, I apologize. And I want to just highlight that within this piece of pancreas, we can really see where all of the different cell types are. This is not a tumor, this is a piece of normal pancreas with precancer, but we can look at all the different cell types and understand where they are, what their relationships are. We quantify at the single cell level close to 1 billion cells in this one piece of tissue. It's just a huge wealth of information that we can really leverage to understand in patients who are in clinical trials what is happening in their tumor in response to the therapy that they're getting. And it's with this tool that we want to really demystify therapy and understand in an individual's tumor, what are the therapies doing? So I want to highlight the importance of doing things in 3D, just so you can understand the general scale of what we're doing. Tissue from clinical trials is often analyzed via tissue microarray. These are, 1 millimeter pieces of tissue that contain hundreds of thousands of cells. What we're proposing here is to analyze billions of cells in a cubic centimeter of tissue. This lets us understand 3D morphology, as well as make sure we have a representative sample and really capture tumor heterogeneity, both with respect to the microenvironment and with respect to therapeutic response. And this heterogeneity is really important, and particularly with reference to the immune system, to the non-neoplastic cells that are responding to the tumor. We've done modeling of pancreas tissue and really seen striking heterogeneity with respect to the immune infiltrate within the same neoplasm, within the same tumor, that suggests that we really need to look at big pieces of tissue in 3 dimensions to really understand what's going on. So when we do this 3D analysis of the tumor that's been taken out in surgery, there's several key questions that we want to answer with this. First is, what are the molecular profiles, excuse me, what are the molecular profiles of the tumor cells that are not responding to the therapy? Is there something about the tumor cells themselves that is allowing them to be resistant? And then what are the cells around the tumor, those non-tumor cells? What is that in the tumor cells that are not responding? How are the immune cells different? How are the stromal cells different? And then finally, what pathways could be mediating the crosstalk between the tumor cells that aren't responding and the cells in the microenvironment, right? The cells all talk to each other. And then once we know these pathways, can these be targeted with novel combination therapies that can be added into the next arm of the trial to overcome this resistance mechanism? This is the tissue design or tissue analysis pipeline that we've come up with. And I put it up here just to highlight that we have multiple different profiling approaches that we can use to understand what's going on in the tumor cells and what's going on in the microenvironment to really understand in 3D, how are these tumor cells becoming resistant to therapy. We also want to do temporally resolved analysis. So looking not at a single time point very deeply, but looking much more broadly over time. And this can help us know, again, what are the molecular profiles of the tumor cells that recur, but also what is the timing of this resistance and recurrence? We have to know when the tumor cells are becoming resistant so we can design regimens that combat the resistance. And also, since we're talking about immunotherapy in many of these trials, what is the evolution of antigen recognition and how the immune system recognizes the tumor? How does that change during therapy? And again, all of this information, the goal is to use it to design new regimens for new trials that combat the resistance that we've seen in the previous trial. So it's an iterative approach. So as I mentioned, we get to look at these things widely over time, we get multiple different samples of tumor in the course of the patient's standard clinical care. So within the trial, all the patients get a pretreatment biopsy. That's our ground zero, our untreated. We get the post-treatment primary tumor when the patient has their surgery, and that is where we do our really deep spatial profiling. And then we get serial blood samples where we can look for circulating biomarkers. And then unfortunately, many of these patients will recur and we'll have access to that recurrent tumor to then look and understand how did the tumor escape the therapies that we tried. And again, we can look at immune regulation as well as how the tumors evolve over time. And I really want to highlight one of the concepts that Dr. Geary brought up, that this really is a partnership with the patients, and we are very fortunate to work with patients that allow us to learn from their tumors so that we can design better therapies. So I want to just close by saying, why breakthrough cancer? Why is this project a good fit? Part of it is strength in numbers. We have 4 of the highest volume pancreatic cancer centers in the country that we can use to accrue the clinical trials. So this is, as others have mentioned, this is Johns Hopkins, Memorial Sloan Kettering, MD Anderson, and Dana-Farber. So we have very high volume clinical expertise in pancreatic cancer to accrue the trials. So these trials that would often take 2 to 5 years if they were at a single institution, we can significantly accelerate that pace. And again, we can learn from every patient at an unprecedented scale so that we can understand in humans that are in the trials how people are responding and becoming resistant to these therapies to then design better trials in the future. And our goal is really broad. It's to build a clinical trial platform that can form a template for the rest of the country. And what we're doing is simply not possible at one institution. Hopkins going to do it ourselves, MSK going to do it ourselves. We really need this radical collaboration across the 5 institutions, as well as between Breakthrough Cancer and Lustgarten, to really push this vision forward. And it's our hope that by understanding science in patients, we can really improve therapy for pancreatic cancer. And with that, I will turn it back over to Andy. Thank you.
Thank you so much, Laura. That was wonderful, and as promised, some really beautiful images. And I hope people really are impressed with how special those images are that you're able to generate, and how much more information you can get when you can really look at the interaction of cells in place and the interactions of different genes in the tissue where they exist, and without having to take it out and dissociate it and chop it up. So wonderful presentations. And again, if Dr. Geary can come back and join us, I hope people really took away those points that we started from, of the importance of collaboration and how that can make things happen faster. How the focus on patients, both from patient needs and learning from patients, really helps us to do better, and how these are teams that are not slowing down, that they really understand the critical nature of the work that they're doing and how the results were needed yesterday, and we're not going to wait and slow down to have them come. So I see there's questions coming through, and we'll get to these questions, but I'll start with one. So because Dr. Wood and Dr. Geary both have been so good about representing as a team and really presenting this as the groups that you're working with and presenting the opinions collectively, I'll give you a moment to step back into your own role as physicians and as scientists, And it'd be great to hear from you kind of what's really exciting to you right now, what's happening in pancreatic cancer research, and how is that influencing your own priorities and your own work in your lab. Kind of, you've both been involved with this for a long time, have seen a lot of things change. Kind of why is right now such a special time for your own work in pancreas cancer? So maybe Dr. Wood, please take it first.
Yeah, I mean, I think for me, I'm a pathologist, which for folks on the webinar who don't know exactly what that means, my clinical role is to look at pieces of tissue under a microscope and make diagnoses. And I think that we're really at a time now where we can do so much with tissue samples from patients, that are obtained as part of clinical care, but we can learn so much from them. And so it's an amazing opportunity to understand cancer as it happens in patients rather than cancer in model systems. And my lab uses model systems too, and they certainly play an important role. But I think being able to understand both pre-cancer and then cancer and then therapeutic resistance in patients so we can really understand how to target it in patients, to me, is one of the really exciting things about the field.
Thanks, and Dr. Gary?
Yeah, thanks for that question. So I'm a medical oncologist, and I'll also run a cancer biology research lab. So my lab has for a long time been interested in the molecular characterization of pancreatic cancer, understanding all of the genes that are altered, either in your DNA at baseline and what we determine the germline. So you're born with that DNA, and you may have alterations in your DNA that may predispose to pancreatic cancer, or in the tumors as they arise in patients. So when tumors arise, they develop mutations in genes or alterations that differ from the rest of the cells in their body, the normal cells in their body. And those genes lead to changes in proteins that really drive the formation of cancer. So for a long time, my lab has been really interested in dissecting that molecular identity of pancreatic cancer and subtyping pancreas cancer rather than just saying it's all one disease that's really bad. What are the different flavors of pancreatic cancer, and can we find subsets that we can target with particular drugs? And for a while, when we didn't have RAS inhibitors, we were looking at very small subsets of the disease. But more recently, with the development of RAS inhibitors, really changed what my lab focuses on. I've for a long time been interested in the biology of RAS, but now we have these really great inhibitors that we can test in patient-derived models that we've taken a sample from a patient, we've grown it in the lab, and we're actually studying the response of that patient's tumor to these drugs or other drugs, sometimes in concert with what the patient's getting treated with in the clinic. And so I'm very interested in this idea of precision medicine, understand the molecular subtypes, model it in the lab, use the newest drugs to see what works, and hopefully lead that to clinical trials that may make a difference for patients.
Thanks, Andy. That's great. And I think we've seen people that pay attention in other cancers, that's really been the difference maker, right? Is when we can start to treat the patient's own tumor. And that's something that's been a little bit elusive in pancreas cancer, but hopefully is changing quickly. And just building on what you said, a few questions that came up in the chat, and hopefully you can just clarify a little bit. I know we talk about, genes and genetic screening and mutations, and we often aren't as clear as we can be about the difference between the mutations that you might inherit from a parent and are present in all of your cells versus the mutations that appear in a tumor and are really driving just the formation of that tumor on its own, but are not inherited. So maybe, could you help to distinguish a little bit the difference between these, inherited germline mutations and acquired somatic mutations, and when and how those are screened for, and how RAS and KRAS and the different KRAS mutations fit into that hierarchy?
Sure. Yes, I'm happy to comment on. Thank you for that excellent question. So a certain subset of pancreatic cancers occur in patients for whom there's a familial predisposition to developing pancreatic cancer. It tends to be the minority of pancreatic cancer. So most pancreatic cancers occur what we'd say sporadically. They occur in the pancreas without an underlying gene alteration in the patient's normal DNA and in the rest of the cells of their body. That subset or in terms of their germline DNA, the DNA that they're born with, it's about 10% of patients for genes we know about, and maybe, globally about 10% of cases where we don't know the gene, but we know there's a family predisposition to pancreatic cancer. So now for all patients, it's recommended that all patients diagnosed with pancreatic cancer undergo germline genetic testing and genetic counseling. So each patient we see, we refer them to a genetic counselor who talks to them about their potential risk for pancreatic cancer based on their family and other cancers that might have occurred in the family, and send off DNA for sequencing and what they do is they look at your DNA and from blood cells or from a swab of the inside of your mouth, and they look for a certain subset of genes. We're up to maybe 130 or 140 genes that we look at routinely. And then that gives some sense, are there mutations in genes that we know can predispose to pancreatic cancer, such as BRCA2, the breast and ovarian cancer familial gene, or other genes? And if we find those, then that doesn't necessarily mean that patient's going to develop pancreatic cancer. In fact, only a minority of patients with those gene alterations actually go on to develop pancreatic cancer. But what it allows us to do is to put patients into a risk category and consider additional screening. And there are great screening programs really pioneered at Johns Hopkins University, as well as elsewhere, that there are certain imaging tests and certain endoscopy procedures that can be considered for patients at risk of pancreatic cancer. So that's the germline DNA risk of developing pancreatic cancer. That's an important consideration in any patient with pancreatic cancer. Interestingly enough, we do not see KRAS mutations in the germline, in patients' normal DNA. And that's a really fundamentally important point, that why don't you see the major driving gene that causes pancreatic cancer? Why don't you see that occurring in the germline and predisposing to cancer? Well, it turns out that mutation is so strong, normal cells probably don't tolerate it well. And it's a different type of mutation, typically a loss of a mutation that restrains cancer, that we tend to see in patients' normal DNA. So we don't see RAS mutations that are inherited for the most part, and we don't see them as a predisposing factor for cancer. That said, now we talk about what mutations or alterations occur in the tumor. So when a patient develops a tumor, a lot of changes occur, in particular in those cells that become the tumor, not throughout the body, but really focused in those cells that go on to become the tumor. And so RAS mutations, that's where they occur in 90% of cases in pancreatic cancer. We also recommend that all pancreatic cancer patients get what's termed somatic DNA testing. So we take a piece of the tumor or a biopsy, a brushing from an endoscopy procedure or a needle biopsy, send that for sequencing, because there are certain molecular features, even not considering RAS mutations, but there are certain molecular features that can help guide therapy for those patients. And the discovery of these RAS inhibitors is going to expand the importance of that even more. Knowing what type of KRAS mutation that the patient has, whether it's what type of mutation, is it a mutation in KRAS or perhaps even another RAS protein, and understanding that and then directing those patients to the right drugs and the right clinical trials.
Can I add something just real quick? I mean, I think, Andy, that was a great overview. The one thing I wanted to add is, when we think about KRAS mutations, they're so common in pancreas cancer. Kind of an obvious question is, why don't we just screen everybody's pancreas for KRAS mutations? And then when we find them, we'll know that person has cancer. And the challenge is that the precancers that Andy briefly mentioned, they have a long name that's very not conducive to these types of webinars, but the abbreviation for them is PANINs. They're precancerous. They'll never hurt the patient by themselves. They're just very, very, very common. Everybody has probably many of them, and they all have KRAS mutations too. So the KRAS mutation by itself doesn't really identify anybody as high risk, because I mean, I can talk about myself. I probably have at least 10 or 20 PANINs, and they all have KRAS. And so you would overtreat folks if you use just the KRAS mutation. So I think there's a challenge in the screening questions of how do we identify not the people that have the precancers, because everybody does, but the precancers that are at highest risk for progression.
Yeah, that's a great point, Laura.
Thank you.
And thanks, Laura, for bringing that up, because I think as always, when we do this, we get lots of questions about, well, what's new in screening, right? And we talk about all the time, early detection of pancreatic cancer is one of the major hurdles for us having the outcomes that we want to need. So maybe, Laura, can you talk a little bit more about kind of what's new, what's happening in terms of really moving towards screening, getting towards an ideal world where we can detect all pancreas cancer early, and maybe some of your other work around really understanding what are those precursors and how do we get our heads around them?
Yeah, I mean, I think there's kind of two separate questions. One is the very clinical question on screening, and I want to highlight there's a consortium here at Hopkins that I'm only tangentially involved with, which is called Cancer of the Pancreas Screening. I think Andy mentioned it as well. And they're really focused on patients who are at high risk for pancreatic cancer, mostly based on family history. What's the best way to screen those patients? And they've shown that in that population, screening does result in earlier diagnosis of cancer and does improve survival, but that's screening based on basically radiographic imaging. So either, CAT scans or endoscopic ultrasound, which is where an endoscope is placed down into the stomach or the duodenum and the pancreas is imaged. So I think we have some sense of who the high risk patients are, based on family history. There's probably other groups that we need to learn more about. And we have some good tests, but we also need better tests and better biomarkers as to who's high risk. As far as understanding the basic biology of precursor lesions, I showed the fancy spinning pancreas video of normal pancreas, and one of my lab's other interests is really using this 3D technology to understand, what are the precursor lesions in normal pancreas and how do they evolve and how do they progress? And so we've accumulated a cohort of close to 50 normal pancreas samples. We've modeled all of the precancers. And again, they're incredibly common. Every pancreas we modeled had many. There's probably hundreds or even thousands in an individual pancreas. So again, highlighting these precancers are really, really common. And each individual one has a very, very low risk. And so I think what we need to understand now is what are the molecular alterations that define those precancers that are at high risk for progression, whether it's changes in the tumor cells themselves, changes in the microenvironment, and then can we pivot those into early detection or screening trials to use those to better identify the patients who have the highest risk lesions.
Thanks, Laura. That's great. I mean, because as you mentioned, you're finding some surprising things when you look at those early lesions. One, that these KRAS mutations are very common. They occur very early. So it may not be enough of what to say, but this kind of approach will help us identify what are those things that are uniquely associated with a precancerous lesion that's going to become pancreas. So we know the right people to treat. And what the CAPS study has shown us, if you put these people in surveillance, they do much better. You find the cancers earlier, they can go to surgery to have much more successful outcomes. Shifting gears a little bit, but another question for you, Laura. One of the powerful things when the way you're looking at the tumors is you're not only looking at the tumor, but the tumor microenvironment, the other associated cells. And we've talked a lot about the stroma, this sort of capsule that forms around the tumor that makes it difficult to treat sometimes. So maybe can you give a little bit more color on what we're learning about the stroma and how to use that in making decisions about the types of drugs to use or the ways to get around that barrier?
Yeah, so pancreatic cancers are really unique, and thank you for that question. They're really unique in that if you took a piece of tissue that was a pancreas cancer, most of the cells in that would not actually be the cancer cells. There's many, many more cells that represent non-malignant cells that are in the surrounding microenvironment. And probably the most prevalent of those cells is a cell called a fibroblast. It's a spindly kind of cell, mesenchymal cell. And we've really had an evolving understanding of the roles that those play. As Andy mentioned, they are a barrier to therapy because they make it hard to deliver drugs. And so early on, one of the thoughts was like, oh, if we ablate the stroma, then the drugs will get in, all the patients will do better. But it turns out that there's different subsets of those cells, and some of them are actually constraining the tumor. So if you just ablate all of them, then the tumors actually grow more quickly. And so there's a lot of complexity to understanding the different groups of cells in the stroma, which ones are promoting the tumor, which ones are restraining the tumor. And it's really been molecular profiling using some of the techniques that Andy mentioned, like single cell RNA sequencing and also spatial profiling techniques that have let us understand the diversity of these cells in the microenvironment, and using model systems try to understand what their roles are. So we know we can't just ablate all fibroblasts, but maybe we can ablate subsets of them that are actually promoting tumorigenesis. And similarly with immune cells, and I am not an immunologist for sure, but I'm fortunate that our team has, Lei Zhang and Stephanie Dugan and other folks from the other institutions who are amazing immunologists who have helped us in designing our trials that way.
Thanks, Laura. And then now maybe another question, sort of pulling out from the questions that are getting submitted with 3 non-immunologists here on the screen.
Oh no.
So one of the plus sides of this very common KRAS mutations in pancreas cancer is that it gives a handle of something that's unique about the tumor cells that's not present in other cells. And I think we heard about using that as a small molecule target that hopefully will be safe to the non-cancerous cells and target them, but also gives a handle potentially to drive the immune system. And we hear sometimes talking about KRAS and vaccine development. So I wonder if either of you could comment a little bit about how the development of vaccines that'll get the immune system to target cells that have mutant KRAS, how that's unique and maybe complementary to these small molecule approaches and kind of where those stand. I know it's neither of your personal work, but maybe you can just shed some color, because I know a lot of people hear KRAS and kind of put it all together, but these are really two.
Yeah, I can very briefly comment, and then I'll turn it over to Andy, who probably knows more than I do. But I will say that within our Demystifying Pancreatic Cancer Therapies team, we have what we've called the Therapeutic Prioritization Committee. And this is a committee of medical oncologists as well as basic and translational scientists who are choosing the agents for the next trial. And I think that's another really important side of the collaborative aspect of these projects, is it's not just, Laura's in charge, we pick Laura's favorite trial. I mean, I don't know anything about trials, so I don't have a favorite trial, but it really is a team effort, and the group kind of comes up collaboratively with what the next trial is. And there has been a lot of discussion of incorporating a KRAS vaccine into one of the future trials for the Demystifying Pancreatic Cancer Therapies project. There's peptide vaccine approaches, which are protein-based, as well as mRNA. So I think that's something that our kind of broader clinical team is really interested in. And again, once it gets into kind of our tissue processing science and patients pipeline, we get to understand a lot about response and resistance.
Great. Yes, I'm happy to comment. So I agree completely about the importance of that committee that Dr. Wood mentioned, and the potential of a vaccine. So the KRAS is expressed. It's a mutant form of the protein. We're talking about targeting that mutant form of the protein with small molecule inhibitors. But as somebody in the chat also asked an important question, well, if that's a mutant protein that's different from the rest of your body, can't you just develop drugs to target that mutant protein? Yes, that is a major goal, that those drugs that specifically target a mutant protein, that's a really important opportunity. Similarly, those mutant forms of the protein that get expressed, well, your body's not necessarily seen those mutant forms before. And so what does your immune system do? It recognizes foreign proteins and viruses and cells in your body, bacteria. So your immune system can also, in theory, and it's been shown in practice as well, recognize mutant forms of RAS that are expressed in cancer cells. And so there's a huge effort to work on leveraging the patient's own immune system to target RAS mutant cells. And one way you can think of vaccines, at its simplest form, take the mutant form of the protein or a DNA vector that expresses the protein, or an RNA vaccine, such as the COVID vaccines that then express the protein, give it to a patient, they develop antibodies and immune responses to the RAS protein, which then hopefully can be directed to the patient's tumor. So there are vaccines. There are also strategies that say, well, if your drug binds covalently or irreversibly to the KRAS protein, that protein can be presented by the cells through normal immune mechanisms, and then you can develop antibodies, or perhaps even what are termed cellular therapies. T cells are the sort of major cells that police the body for foreign viruses and bacteria and other types of invaders. You can turn those T cells directed at the mutant form of the protein that's actually bound to drug. So you've created a new way to present cancer cells to the immune system. And then there are cellular therapies. Can you take T cells from outside the patient's body and direct them at the RAS protein? There's been really interesting proof of concept data that's been published in the literature recently. The idea of taking a patient's own cells, engineering them and putting them back in to target the KRAS mutant tumors. And then there's a whole host of companies that are focused on making what we term off-the-shelf therapies, T cell therapies that could be given to multiple patients that are directed at RAS mutant tumors. And so there's an enormous amount of effort directing the immune system to KRAS mutant tumors. And I also see there's a question in the chat about, can you create an adaptive living therapy drug? Really interesting concept. In theory, that's what your immune system could be. If you could direct the immune system to a RAS mutant tumor, your immune system can adapt and hopefully with the tumor. So really, really interesting questions.
The one point I'll add is that, again, from a non-immunologist, there's lots of ways that pancreatic cancer manages to evade the immune response and create an immunosuppressive microenvironment. So it's not going to be enough to just do a KRAS vaccine. We also have to overcome all these other immunological hurdles. So it really will be, I think, a combination of multiple vaccine and multiple immunomodulatory agents that allow us to, develop the KRAS specific immune response, but then actually get it into the pancreas cancer.
Yeah, that's a great point, Laura, that any one treatment isn't going to be enough. And there's a lot of effort to think about how do you use small molecule inhibitors in concert with immune therapies, and how do you sequence them. And just as there can be resistance to these drugs, these small molecule inhibitors, there can be resistance to the immunotherapies. So understanding that as well.
Yeah, and I know we didn't talk a lot about it today, but just so folks are aware, these are things that not only are potentially useful, but are being tested now and showing some promise, including using vaccines plus other immunomodulatory agents after surgery to prevent recurrence. And now, very excitingly, the first trial started using vaccines in people that are at high risk to show if there can be an immune response and hopefully build up to something that can be used to lower risk for developing pancreatic cancer in the future.
And one of the exciting parts about that concept is in the precursor lesions, maybe some of these immunosuppressive mechanisms aren't as well developed. And so it may be less of a hurdle to get the immune system to recognize those. So that I'm super, super excited about that.
Right. So I know that there's a lot of questions and they keep coming in. And I know we haven't gotten to all of them. We'll try to answer some of them offline if it's possible, when we're coming up on time. And I do want to give one last question and one last chance for both Dr. Agari and Dr. Wood to give the answer. And I won't ask it as if you're successful, but when these projects are successful, kind of what does success look like for you? And I think both maybe in the shortish term and in the long term, what would this really mean in your clinical practice, for your patients, for families down the road? Maybe we'll start with you, Dr. Agari.
Yes, excellent question. I think, that we have two major chemotherapy regimens for pancreatic cancer patients, and giving the right one up front makes a big difference. And if you respond, you get a benefit. If you get the next chemotherapy regimen after the first one, you're less likely to respond to it than you were if you got the right one up front. So that's a critically important limitation of our therapies, is that we only have a couple major chemotherapy regimens. So short-term success, even with a low bar, could be adding another approved therapy for use after chemotherapy that would give patients perhaps additional response, an additional quality of life and duration of life, even if measured on the order of months, that would be a major advance to take a well-tolerated pill, even in later lines of therapy. I think in the long term, viewing these RAS inhibitors as the foundation upon which we'll build, and perhaps not giving them in the later lines of therapy, but moving them forward and giving them early on in therapy and giving the right combination of agents with a RAS inhibitor to provide more durable and longer term responses, and eventually one day perhaps even a cure, or make pancreatic cancer a controllable disease that we can live with, manage, adapt to resistance mechanisms, give additional therapies, and really lead to, I think, major improvements in people's lives. So that would be my hope from this project, that this is the beginning of a long-term effort, sustained focus on KRAS and using these therapies effectively, and ultimately making a major impact for patients.
Thanks, Dr. Gary. Dr. Wood?
It's a hard answer to follow. It was very, very well articulated. I think I'd share Andy's long-term vision of, making pancreatic cancer a long-term treatable disease, and that we have multiple tools in the toolbox. We have multiple different types of therapy, and we understand who to use those therapies in, right? Who's going to respond to what. And then when they become resistant, we know what the next step is. And again, to improve survival and improve also the lives of patients, right? The current chemotherapy regimens are very challenging and very toxic. And if we can have less toxic chemotherapies as well, I think that's a huge win. I think for the shorter term in our project, one of our main goals is to really develop this pipeline for clinical trials in human tissue and biosample analysis that will lead us to that longer term vision, right? We need to understand how pancreatic cancers respond and how they become resistant. And to me, that's one of the key tools in the toolbox that will let us get to this vision of having multiple therapies, multiple things to do once patients recur. And so I think just developing this strategy for doing science in patients, for understanding, using human clinical trials to really understand how pancreatic tumors occur and behave in real humans. I think that's our short-term goal.
Well, thank you very much. And I think I'd say for myself, and I hope everyone else on the call here, we all share your vision for where we need to be and where we should be in the future, and making pancreatic cancer a treatable and a curable disease. And I feel much more hopeful that the path to get there is achievable with folks as smart and as dedicated and as humble as yourselves, helping to lead us there. So thank you so much for joining us today and sharing your very impressive work and the importance of this partnership between LustGarten Foundation and Breakthrough Cancer to really address these most critical questions in pancreatic cancer. Thank you very much. So Linda, I'll turn it back to you to give us some final words and to close this out.
Great. Thanks, Andy. And thank you, Dr. Gary and Dr. Wood. And I deeply hope that to everyone who is watching this and will watch it later, that you see our deep bench of scientists and their deep commitment to transforming pancreatic cancer into a curable disease. And you can see the deep science and thinking that is going into the work that we're funding in partnership with Breakthrough Cancer and with all these amazing institutions. But before we say goodbye today, please take a look in your emails. You'll be receiving a survey today, and we would really appreciate you completing that survey. Also, check your email tomorrow for details about a super exciting LustGartenLIVE coming up March 8th in celebration of International Women's Day. We are so honored to be able to invite you to meet some of the rising stars of pancreatic cancer research. You'll also hear from our 2022 Ruth Bader Ginsburg Career Development Award recipient, and that will be followed by a panel comprised of some of the luminaries of pancreatic cancer research. You won't want to miss this, so please sign up, and we'll see you March 8th, 11:30 Eastern Time. And thank you so much for attending this LustGartenLIVE and for your commitment to the LustGarten Foundation and to pancreatic cancer research. Thank you all.